If we imagine a mosaic composed of countless tiny, colorful tiles forming a vibrant and intricate picture, each tile represents a fragment of genetic material from organisms in the environment, and when combined, they create this beautiful mosaic that represents the biodiversity of the ecosystem.
eDNA stands for environmental DNA, and environmental DNA is fragments of DNA that comes from organisms that live in the water column, in our case. The process starts in the field where we take our water samples and then filter them down so we can mail them back to our lab here.
Once we get to the lab here in Fairbanks, we extract the DNA from those filters that we collected in the field. We PCR amplify those barcodes or gene regions that we've targeted. We send it off to a sequencing center.
We decided to use eDNA to try and answer our research questions because eDNA will capture anything and everything that is in the environment. We have to figure out if it's also an accurate way for us to collect our samples. So one of the things we're doing is comparing our eDNA samples to other conventional sampling methods that we've been using, such as beach staining for fish and percent cover in intertidal areas.
If we can see predictable patterns in species composition across the tree of life along these glacial gradients as they're changing from glacial fed to rainfall fed, then maybe we can make predictions in the future about how entire communities of organisms are going to change as glaciers continue to recede. The scale of this project is really cool because we are analyzing tiny segments of DNA that we collect from the water, and those segments of DNA represent a ton of different organisms across the whole tree of life, from fishes to invertebrates to algae and even mammals and seabirds. And so by piecing together all of these tiny fragments that we actually can't see with our own eyes, we can answer these really big picture questions on how entire communities and ecosystems are changing across these glacial gradients.